It's a great privilege to introduce Anne Bertolotti today as our keynote lecturer. Anne is a fantastic colleague and a community member. Her brief career is Strasbourg studies, PhD with Pierre Chambon on transcription, moving to NYU, where I just left. So we kind of overlapped in a zero time, but 1 year after another. And she did a very nice postdoc with David Ron, getting involved now in protein folding, misfolding, and proteostasis. And that was something which she inherited in her independent work. Returning back to Europe, she started her independent work in Paris, in France, staying there for a while, but then getting lured to this fantastic jewel of European size, LMB Cambridge, where she is since 7 years, I believe. And today we are going to hear a little bit about her part of the work in the lab, which talks about proteasome homeostasis. But her major interest in the lab is understanding the regulation of protein folding, misfolding, and the ways how the cells fight against that to prevent the neurodegenerative diseases. As I started the talk, I said Anne is a fantastic colleague, always happy, always motivating, and she's also an incredible sportswoman. I fitnessed that by skiing with her. Actually, I was on snowboarding, and she was skiing, and we had an amazing time. And then here in Cold Spring Harbor, I was just deciding to run with Brenda, and somehow Anne just came along telling us that she wants to run with us. And both Brenda and myself, we were terrified, because Anne is known as a very polite competitor, but running always faster than others. And that politeness makes her that people don't notice how competitive she is. So we'll see the movie now. Can I get the next slide? Here is the way. Look at this, look at this.
Did you get a video of that?
What do you think?
Yes.
So that's this politeness, and I think in science this is also required, because if you win by politeness and smile, it's nicer. So welcome, Anne, and welcome to the meeting.
I've dropped the microphone on the way, running to the stage. I was worried that I needed to be first. Right. Yeah, it's a real treat each time to be invited to join the Ubiquitin family. I think there's a true community spirit in the ubiquitin field, and I enjoy it a lot. I've had a wonderful week, and there's more to come. I'm really excited by the rest of the program. So I'm also honored to have the Cold Spring Harbor floor for longer than event today. And so I've decided to do something special. Rather than giving you an overview of diverse aspects of protein quality control we work on in the lab, I thought I would use this unique opportunity to focus on the proteasome, and the question I will ask is, can we survive when the proteasome fails? So we've had wonderful talks about the proteasome at this meeting. Andy Martin and Andreas Matuszek earlier told us about the proteasome. The proteasome is really a key component of our protein quality control systems. The proteasome degrades a broad range of cellular proteins and thereby controls essentially all aspects of cell and organismal function. And consequently, obviously, proteasomal degradation is absolutely vital, and a consequence of that is that failure of proteasomal degradation has been suspected to be associated with a broad range of human diseases, including the devastating neurodegenerative diseases, which are caused by accumulation of proteins that should otherwise be degraded, and I'm sure many of you remember the seminal work of Juan Capito in this area. So the idea we entertain in the lab is fairly simple, right? If failure of proteasomal degradation is at the origin of a broad range of diseases, perhaps we can try and think about ways to rescue this defect, with the view that perhaps if we manage to do that, this could pave the way for the development of strategies that could help preventing or perhaps treating diverse diseases. So don't get your hopes too high. We haven't cured anything at this point with this approach, but this is the idea in the back of our mind that motivates the work we do, and that motivates us to look for approaches that can help cells surviving when the proteasome fails. So in the lab, what we do, that's our fast-track modeling for complex diseases. We block proteasomal degradation, and we do that in different ways, genetic or pharmacological inhibition, and this inevitably kills cells. And then this provides us with a really robust experimental system that we can use and ask, can we find ways to survive these otherwise catastrophic conditions? And in today's talk, I'll review for you in a sort of integrated way the different approaches we've discovered in the lab to help cells surviving when the proteasome fails. And these approaches have actually shed light on fundamental aspects of proteasome biology. So, as you all know, proteasome inhibition kills, and this property has actually found its way to the clinic because, as you know, proteasome inhibitors are used for the treatment of multiple myeloma. But curiously, it wasn't clear why proteasome inhibition actually kills. So you all know that when the proteasome is inhibited, cells accumulate loads of proteins that should be degraded. But there's another aspect to proteasomal degradation which curiously hadn't been much considered. By degrading proteins, the proteasome inevitably recycles amino acids, right? And so we wondered whether proteasomal degradation was not only important to degrade proteins that need to be degraded, but also in maintaining amino acid homeostasis. And I must acknowledge that this line of thought was actually very much inspired by the work that Yoshinori Osumi had done on autophagy. Before autophagy was recognized as a major degradation system, remember that autophagy was discovered as a defense against starvation, right? So autophagy is induced in response to starvation, and in this way, cells recycle intracellular components to provide nutrients, and that enables cells to survive under starvation conditions. So consequently, autophagy encoding genes are absolutely essential for yeast cells and mammalian cells to survive under starvation conditions, but otherwise dispensable. In contrast, all of the proteasome subunits are absolutely essential genes. You can't survive without proteasomal degradation, and this under physiological or non-physiological conditions. So to me, this was the strong motivation for examining whether proteasomal degradation was important in maintaining amino acid homeostasis. And so we asked a very simple question: what is the consequence of proteasome inhibition on amino acid levels in cells? And we asked this question in diverse experimental systems. The first one was a yeast system in which we took advantage of a thermosensitive mutant harboring mutation in one of the AAA subunits of the proteasome, RPT6. And as a result of this thermosensitive mutation, when you switch cells, when you grow cells to the non-permissive temperature, the proteasome can't function and the cells dies. And this mutant is extremely robust. As you can see, it's not a leaky mutant, and it's so very useful to use as a model of proteasome impairment. So we took advantage of this mutant and asked, what's the consequence of proteasome inhibition on amino acid levels in the cells? And we found that following 5 hours after proteasome inhibition, we saw that the levels, the abundance of pretty much all amino acid had decreased in the cells, suggesting that inhibition of proteasomal degradation depletes the intracellular pool of amino acid. And then the big question was, and so what? Is this important? Does this have important consequences for cell fitness? And to try to answer this question, what we reasoned that if this decrease in amino acid was deleterious for cells, then perhaps we could rescue some defects following proteasome inhibition by adding back amino acids, right? So we then looked at whether amino acid supplementation could protect cells from following proteasome inhibition. And so proteasome inhibited cells, after here 20 hours following proteasome inhibition, will be all labeled by propydium iodide, indicating that these cells are actually about to die. But if the cells had been cultured in the presence of additional amino acids, and bearing in mind that these cells are grown in the normal rich media, but we supplemented with an additional supply of amino acid, you see that they were not labeled at all with propydium iodide. This was really striking because it suggested that supplementation with amino acid protected cells from cell death resulting from proteasome inhibition. And this was so striking that we were not satisfied with this result, and we wanted to really check whether these cells are actually viable. So we returned these cells to the permissive temperature and asked, well, if these cells are viable, they should give rise to colony, okay? And so these are the cells before proteasome inhibition. They are viable, they give rise to colony. Proteasome inhibited cells, if you inhibit the proteasome for about 20 hours, the cells, virtually all the cells die. But if the cells had been supplemented with amino acid, you can see that they are protected. So there's a very, very potent protective effect here of amino acid supplementation. And I want to stress that although amino acid supplementation protected cells from the lethality resulting from proteasome inhibition, these cells did not divide. And they do not divide because obviously they need a functional proteasome to go through the cell cycle. All right, so here by adding back amino acid, we protect cells from dying following proteasome inhibition. And then the next question was what happens to proteins that accumulate when the proteasome is inhibited. And you can see, so here we look at polyubiquitinated conjugates, and what was striking is that cells accumulated virtually the same amount of polyubiquitinated conjugates, whether they had been supplemented with amino acid or not following a proteasome inhibition. And that was striking because it implies that the load of polyubiquitinated conjugates can't predict cell fate, actually. These cells are about to die, and these cells will be actually viable. So in other words, we've learned here that amino acid supplementation rescues the viability of proteasome-inhibited cells, but this does not affect the load of non-degraded proteins. So what's happening then when the proteasome is inhibited? What's the deficit that is resulting from this amino acid scarcity that follows proteasome inhibition? Well, amino acids are the protein building block, right? So we wondered whether this decrease in amino acids following proteasome inhibition had deleterious consequences on protein synthesis. And indeed, this turned out to be the case. So following proteasome inhibition, you see that there's a marked decrease in protein synthesis. And again, I'm stressing that these experiments are done under rich conditions, so arguing that this intracellular recycling of amino acid is very important to sustain the rate of protein synthesis. But so there's this decrease following proteasome inhibition, which is rescued to nearly wild-type level when we add back amino acids. So this had been done in yeast, and of course the function of the proteasome is evolutionary conserved. So we wanted to see whether what we had seen here in yeast also was conserved to higher eukaryotes. And so we examined the consequences of proteasome inhibition in diverse other systems. Here we are looking at mammalian cells, and as we had seen in yeast, proteasome inhibition decreased. In this case, it was only a subset of amino acids that were decreased upon proteasome inhibition, and we rescued the viability of proteasome-inhibited cells by adding back these key amino acids, and the rescuing effect occurred without affecting the load of polyubiquitinated conjugates that accumulate in proteasome-inhibited cells. And to some extent, we observed similar results in flies. So cells can survive with their rubbish, if you like. They survive with their non-degraded proteins, but really the amino acid insufficiency that results from proteasome inhibition is detrimental. So this is as if these proteins that accumulate in proteasome-inhibited cells become deleterious to cells because they accumulate, because they sequester a pool of amino acid which should otherwise be recycled. So then, having observed that proteasome inhibition causes a decrease in amino acids, and of course we were aware that cells have multiple ways to defend themselves against starvation, we then reasoned that amino acid scarcity resulting from proteasome inhibition might be causing a stress to cells, and indeed this turned out to be the case. So in response to this amino acid scarcity, cells adapt to this problem in the canonical way. They induce the integrated stress response. So the integrated stress response has been described and discovered by collective effort of many labs, from its evolutionary conserved from yeast to mammals. And this response consists in phosphorylating EIF2 alpha, a translation initiation factor, to slow down protein synthesis. And this, in turn, will reduce the consumption of amino acids, and that's beneficial. It's an adaptive response to conditions where the supply of nutrients is scarce. And in parallel to this, cells also, in response to starvation, as we know from Osumi and colleagues, canonical response to starvation is to induce autophagy. So many labs, and I couldn't possibly cite them all here, have seen before us that proteasome inhibition induces autophagy, but the key question for us here was how? What are the molecular signaling events between proteasome inhibition and autophagy induction? And having found that proteasome inhibition leads to a decrease in intracellular amino acids, and knowing that amino acid starvation is a signal for autophagy, obviously we wondered whether this decrease in amino acid might be the signal that leads to autophagy induction when the proteasome is inhibited. And this turned out to be the case, and this signaling occurs through the canonical signaling pathway that has been largely elucidated by the Sabatini lab, involving the RAG GTPases upstream of mTOR. So I've shown you that proteasome inhibition depletes intracellular amino acids, and cells try to adapt to this condition and restore amino acid homeostasis by inducing two adaptive responses, the integrated stress response and autophagy. And if these two responses fail to restore amino acid homeostasis, the cells will die unless they are supplemented with amino acids. So this work really tells us that the proteasome is not just a cellular trash can, but it's actually very important as an amino acid recycling machine. As you've heard at this meeting, that this idea has been seen now in other systems. Bacteria also rely on amino acid recycling by the proteasome, and following the work I'm just presenting here, there's been other perturbation of the ubiquitin proteasome system that have been reported to also perturb amino acid homeostasis. And then the question is, does this make sense? And I think, yes, it really does make sense if we relate this to quite old studies, actually, where the total amount of protein synthesis made by the human body have been measured. It turns out that the total amount of proteins that we humans make daily is provided by only a small amount of amino acid that we get in our diet. Or, in other words, our dietary supply in amino acid covers for only 20% of the proteins we make daily, arguing that 80% of the protein we make every day come from recycling. And I would argue that under physiological conditions where we are well fed, just like here after a week at Cold Spring Harbor, a large fraction of this recycling comes from proteasomal degradation. So these new insights into the function of the proteasome led us to wonder whether there were some other aspects of proteasomal degradation that remain to be discovered. And this was a motivation for embarking on genetic screening yeast, looking for suppressors that can, genetic suppressors that can promote cell rescue, cell viability when the proteasome is inhibited. So we continued working on this robust proteasome mutant and asked, can we find genes that when overexpressed rescue the viability of these proteasome impaired cells? And it's really an old-fashioned genetic screen, but it worked really well and we found something new with it. So the first two things we found were not new. One of the suppressor of the screen, very potent suppressor as you can see, encoded the wild-type allele of RPT6. So that's good, that's what you would expect, and we had about 50 clones, independent clones encoding RPT6, showing that the screen was perhaps saturating. The second very potent suppressor of the proteasome mutant was RPN14, and this is a known protein with a known function. RPN14 is a proteasome assembly chaperone, and we heard about the assembly of the regulatory particle of the proteasome yesterday in Seo-Young Park's talk, and I'll just remind you here how this works. And this is the assembly of the regulatory particle of the proteasome has been elucidated over the past 9 years or so through collective effort involving Keiji Tanaka, Saeki, Murata, Ostrasser, and Perosh, as well as Dan Finlay. So the hexameric ATPases of the regulatory particle of the proteasome assembled from 3 modules, each composed of a pair of ATPase bound to specific chaperones. And we've heard about one yesterday, NAS6. There are 4 with similar function, NAS2, HSM3, RPN14, and NAS6. These chaperones bind to the carboxy terminal tails of ATPases to prevent their binding to the core particle until the ring is complete. So in our screen with the RPT6TS mutant, we isolate RPN14, and this makes sense because this is the RPT6 chaperone. And in addition to these 2 known proteins, we isolated a suppressor which encoded for a protein that had not been characterized before. And to me, this was very exciting because now not so many proteins these days for which the function remains unknown. So I thought this was going to be a great opportunity to figure out what was the function of this protein. At that point in time, Ariane, the student who discovered ADC17, sort of panicked. She said, well, actually, there's no function known for this. There's no domains. What am I going to do? And I said, well, just look at this. You know, the suppression is so potent, we're going to find out how this works. And so we took it one step at a time, one question at a time, and eventually, 3 years later, Ariane found what was the function of ADC17, which I'm only summarizing here. So ADC17 binds to the amino terminal domain of RPT6, and this interaction somehow renders RPT6 competent to engage in a productive interaction with RPT3, and this leads to the formation of the RPT6 ATPase pair. And it turns out that this step in proteasome biogenesis appears to be rate limiting. So it's the commitment step in proteasome assembly, so it's not too surprising that it is regulated. So this was interesting, but to us it became even more interesting when we noticed that the expression of ADC17 is regulated, and actually ADC17 is increased massively upon all sorts of conditions that cause accumulation of misfolded proteins in the cells. And that suggested that perhaps ADC17 had evolved to protect cells against proteasomal challenges. So by that I mean that when cells are exposed to overwhelming demands in proteasomal degradation, they have the ability to adapt by making more proteasomes. And we know that there's an increased expression of proteasome subunits upon all sorts of proteasomal stresses. And while this is necessary, this is by no means sufficient, because we've learned over the past 9 years or so that proteasome assembly is a very complex process. So these subunits need to be faithfully assembled into a proteasome complex for this response to be effective. And ADC17 plays a role in making sure that cells can make more proteasome when they need more proteasome, and this is vital because cell viability is compromised when ADC17 is deleted. So this told us that proteasome assembly is actually a regulated process, and increasing proteasome abundance to match the needs is vital. So then that implies or suggested that there ought to be a pathway regulating proteasome abundance. And in order to try and figure that out, we followed ADC17 as a sort of sentinel, reasoning that if we understand what is the pathway regulating ADC17 expression, perhaps that will give us some insights into the mechanisms that are controlling proteasome homeostasis. So we, of course, first started by looking at the usual suspects, so one being RPN4, which is a transcription factor discovered by Alex Warshawski, and there's been lots of work on this, following on the Warshawski work by Dan Finlay as well as Muratalab. RPN4 is the transcription factor which controls expression of all proteasome subunits. So an obvious question was, is RPN4 responsible for ADC17 expression? And the answer is no. We deleted RPN4 here, and you can see that ADC17 not only is present, but it's actually expressed at higher levels than in the control cells. So that told us two things. One is that RPN4 is not directly regulating ADC17, and two, that these cells that lack RPN4 are actually compromised for their proteasomal degradation, and they try to adapt by increasing ADC17, providing another line of evidence to suggest that ADC17 is an important player in adapting cells to proteasome challenges. Next suspect we examine is the unfolded protein response. Tunicamycin is a canonical inducer of the UPR. It induces, it's a robust inducer of ADC17, and so we deleted UPR genes, and we found that ADC17 expression occurs to wild-type levels. So clearly, ADC17 is a regulated protein, but its regulation occurs without involving the usual suspects. So there's something unknown here that we were keen to elucidate, and so we embarked on sort of detective expedition, as we like them, knowing nothing about ADC17 and trying to figure out what regulates the expression of this, hoping to find about mechanisms maintaining proteasome homeostasis. And the detective in this work is a great postdoc in the lab, Adrian Russo. So first, Adrian examined known data sets to look for possible genes regulating ADC17. Most of these data sets involve transcriptional regulation, and for a large part, this expedition was frustrating because all the suspects had very little, when deleted, had very little effect on ADC17 expression, except perhaps SET2, which when deleted slightly increased ADC17 expression, but SET2 is a methylase, it's a general transcription regulator, so we didn't learn much here. But amongst the possible regulator, Adrian found one, SFP1, which when deleted completely, the cells lacking SFP1 are incapable of inducing ADC17. So that provided a starting point to try to figure out what is the pathway regulating ADC17. So what is SFP1? SFP1 is a stress and nutrient sensitive regulator of cell growth. Upon optimal growth condition, SFP1 is in the nucleus where it positively regulates expression of ribosomal proteins, but upon stress, SFP1 relocalizes to the cytosol. So if I tell you that SFP1 is regulated by growth and stress, you won't be surprised to hear that actually SFP1 is activated by TORC1, and this has been found in the lab of Lewis as well as Shore, but we can recapitulate this regulatory loop in our lab. So TORC1 positively regulates SFP1, and in turn, SFP1 negatively regulates TORC1. So that suggested to our detective here that SFP1 could potentially regulate ADC17 by two possible routes, either a direct route or an indirect route via TORC1. So this was the model, and in a first to gain some insights into this regulation, first Adrian looked at the localization of SFP1, and he found that when ADC17 is induced, SFP1 is largely in the cytosol, arguing that the mechanism by which SFP1 regulates ADC17 is likely to be indirect. And then the next question was, does this occur through TORC1? So this was the reason why Adrian tested whether rapamycin induced ADC17. So sure enough, rapamycin inhibits TORC1, and this led to a really robust induction of ADC17. So before going further into continuing exploiting this idea, we really wanted to verify this observation by an independent route, and in my mind, nothing beats genetics, so we turned to a genetic inactivation of TORC1, and so here we took advantage of a TS mutant of TORC1 harboring a thermosensitive, a TS mutant of TORC1, so it's a Hog1 mutant. Hog1 is the yeast homologue of Raptor, and this mutant, as we'd seen with rapamycin, also induced ADC17. So through two lines of evidence here converged in showing that inhibition of TORC1 induced ADC17, or in other words, TORC1 restricts ADC17 abundance. So I've told you in the introduction that ADC17 is a proteasome assembly chaperone. I'm showing you that TORC1 restricts the abundance of ADC17. So that brings the question whether TORC1 somehow regulates proteasome abundance. And so Adrian tested this idea, and he found that rapamycin, similarly to what he had seen before with tunicamycin, rapamycin increased proteasome abundance, and the increase we see here is about twofold, and that's been seen actually to a similar magnitude. We heard about this yesterday in Andreas Matuszek-Stoke. So TORC1 inhibition increases proteasome levels, so this shows that inhibition of the central stress and growth controller TORC1 induces ADC17, and this in turn increases proteasome abundance. So that brought then the question, because TORC1 obviously integrates multiple signaling pathways, so that brought the question, what is downstream of TORC1, and what's the pathways downstream of TORC1 responsible for ADC17 induction? So again, we turn to detective work here and examined available clues. We knew from our earlier work that ADC17 is required to survive tunicamycin stress, and we also knew that this regulation is independent of the UPR. So Adrian went looking for genes that had similar properties that were induced by tunicamycin stress, but in a UPR-independent manner. So that got us interested then in MAP kinases, because two of them were described for the importance for yeast for survival to tunicamycin stress, Hog1 being important and Mpk1 being absolutely essential. And so we looked at these cells and examined whether these MAP kinases were important for induction of ADC17. And it turns out that we observed a perfect correlation between ADC17 induction and ability to survive tunicamycin stress. Mpk1 deleted cells are completely unable to induce ADC17. They are completely unable to survive in tunicamycin. And Hog1 deleted cells are compromised for ADC17 induction and growth in tunicamycin, and we observed similar relationship between ability to survive in rapamycin and to induce ADC17. I'm mentioning here MAP kinase pathway, so before you ask, the question became, well, do the kinases upstream of Mpk1 are also involved in ADC17 expression? And the answer is yes. So here we have isolated a signaling pathway from TORC1 and Mpk1 regulating ADC17, and the next big question was, is this important to maintain proteasome levels? And the answer is yes. So in cells deleted of Mpk1, you can see that these cells are completely unable to adapt to proteasome challenges. They are unable to make more proteasome when they need more proteasome, and that explains why they can't live, they can't survive proteasome challenges. This reminded us of the defect of ADC17, but when we compared the magnitude of the defects of the two mutants, obviously the MPK1 deleted strains had a much more pronounced defect than ADC17. And that then brought the idea that perhaps other factors were involved in under the control of MPK1 to adjust proteasome abundance and to cope with stress. So that then led us to look at the regulation of other proteasome assembly chaperones, and I'm very grateful to Mark Strasser, who provided us with antibody to study this. And what Adrian found was that all the 19S assembly chaperones, NAS2, NAS6, HSM3, and RPN14, all of these are upregulated upon stress in the same way that ADC17 was regulated, and the induction of all the 19S assembly factors, also known as RAC, follow the same paradigm as what I've shown you for ADC17. It's independent of RPN4. It occurs to wild-type level in RPN4 deleted strains, but it requires, it's downstream of TORC1, and it requires MPK1. The cells lacking MPK1 can't do this at all. And so this suggested that one of the important functions of MPK1 is to regulate proteasome assembly chaperones to help cells coping with overwhelming demands on proteasomal degradation. And Adrian wanted to check that genetically, and he suspected that he could rescue the defect of the MPK1 deleted cells by overexpressing the 19S assembly chaperones. I really thought this had no chance to work, but he was very keen in trying out anyway, and he proved me wrong, which is shown here. So Adrian found that expression of the 90-days assembly chaperones could rescue the growth defect of the MPK1 deleted strain. So that is, I think, a very strong argument to showing that one of the important functions of MPK1 is to regulate proteasome homeostasis. And whilst doing this, we also noticed that proteasome subunits, the levels of proteasome subunits also increased in response to various stress, and that was suspected from the work of Warshawski, Finlay, and others. And not surprisingly, this is dependent on RPN4, but interestingly, we noticed that this is also dependent on MPK1. And cutting a long story short, it turns out that there's two levels of regulation for proteasome subunits. One is transcriptional, involves RPN4. One is translational, and that involves MPK1. And the 19S assembly chaperones are under the translational control of MPK1. And this concerted increase of RACs and proteasome subunit controls proteasome abundance and viability upon various stresses. So I know it's a ubiquitin meeting, and you all know that ubiquitin is very important for a very important level of control for protein degradation. But here we wondered whether proteasome abundance was also important in controlling the rates of protein degradation. And so we tested this idea using a variety of proteasome reporters and a variety of assays. I'm just going to show a couple here. So in response to proteasome impairment, when proteasomal degradation is impaired, cells accumulate polyubiquitinated conjugates, and we see a mild defect in MPK1 deleted cells upon basal condition, but this is aggravated upon stress, and showing that the MPK1 deleted cells are indeed impaired in proteasomal degradation, and we validated that using a variety of proteasome reporters. Here I'm showing TS mutant of URA3, which is degraded by the proteasome, stabilized in the proteasome mutant, also stabilized in MPK1 deleted cells. So these cells have a defect in proteasomal degradation because they fail to maintain proteasome homeostasis, and we validated this using different reporters here, showing the famous CPY star reporter that we got from Dieter Wolf, also stabilized in MPK1 deleted cells, as well as the cytosolic counterpart. This is an ERAD substrate. The cytosolic counterpart is also stabilized. So all this shows that MPK1 is important to maintain adequate levels of proteasome, which are required to sustain protein degradation and cell viability under challenging conditions. When we performed this study, I was under the influence of the Baumeister paper that has shown that under normal conditions, only 20% of the proteasome appears engaged with a substrate, but we've heard yesterday from Andreas Matuszek that it may be that the proteasome operates at capacity. So here we've shown you the importance of this pathway to maintain proteasome homeostasis under stress. I think it may well be that this pathway is also important under physiological conditions to fine-tune the abundance of the proteasome to match the needs. So I've shown you also that survival depends on adapting proteasome abundance to the rising needs. And I'm going to perhaps speed up a bit, but we found that this pathway is evolutionarily conserved in mammals. We can inhibit TOR with TORC1 selectively with TORin1, and we saw, as we had seen in yeast, an increase in proteasome subunits as well as the 19S assembly factors, and this leads to an increase in proteasome abundance. And what was interesting here is that this increase is transient, and I think this is in line with all we know about stress responses. They need to be reversible, and at the time we didn't know, but we suspected that the return to basal level could occur by means of autophagy, and there was a very nice poster yesterday by Jerome Roloff's lab showing precisely this. And I think it's very important. It shows that it's very important to maintain the right levels of proteasome. And in mammals, it's presumably ERK5 has been proposed to be the functional analog of MPK1, because ERK5 can rescue the MPK1 deletion, and we found that ERK5 also controls 90S assembly factors. If you knock down ERK5, you reduce abundance of 90S assembly factors as well as the proteasome. So this shows that there's an evolutionary conserved signaling pathway which sort of acts as a switch to increase proteasome assembly and abundance upon demand. And it was very interesting to find that actually this proteasome assembly and abundance is really integrated with growth and cellular metabolism. So TOR, as you know, integrates growth and stress signaling. Activated TOR, it was known that activated TOR stimulates anabolic processes and represses catabolic processes. It had been long known that TORC1 restricts, represses autophagy. I think it's not surprising to find that in a concerted way it also restricts proteasome abundance. I told you that the proteasome is important in maintaining amino acid homeostasis, and you know that TORC1 senses amino acid abundance. So it's not too surprising then that TORC1 also controls proteasome abundance. And as a treat for you who have survived my talk, I have a short animated movie to present that will sort of summarize everything I said in 1 minute. Here we go. This is the first time I'm presenting this. I just got it this morning, so let's see how it goes. Right. If I manage to turn it on. All right. So under optimal conditions, cells are busy making lots of proteins that are required to perform all aspects of cellular function. And for this to go well, you need efficient quality control in the cells. You need chaperones, these little hands in orange here, which make sure that proteins are folded properly, that they don't aggregate. Sometimes we make errors, but errors are dealt with, and misfolded proteins will be degraded And for that, we have the proteasome. We have autophagy as well, but I'm just showing the proteasome on this movie. Proteasomal degradation is not only important to remove proteins that need to be degraded, but also in recycling amino acids, which are important to sustain vital rates of protein synthesis. And all this goes well, but upon stress or in diseases, misfolded proteins accumulate, and cells have defense mechanisms and have learned to survive this by multiple ways. And I've shown you one way here today, which consists in increasing proteasome abundance to resolve the protein misfolding stress. This is under the control of mTOR, and the way this works, it's a translational regulation that increases expression in a coordinated manner of proteasome subunits as well as assembly factors. This increase in proteasome abundance will resolve the stress, get rid of misfolded proteins, and when the conditions when we return to optimal conditions, then proteasome levels will go back to normal. And we've learned from Jeroen's lab that the way proteasome levels return to basal level is mediated by autophagy, and it looks like it's not bulk autophagy but some specific form of autophagy, so it's likely that there are some more regulatory elements that remain to be discovered. With that, I'll thank the people in the lab that have done the work. Amila was involved in the amino acid homeostasis work. Ariane discovered ADC17. The function of ADC17 was elucidated by a team effort with Ariane, Adrian, Zen, and Anna. And Adrian did all the work I've shown you today on the TOR control of proteasome abundance. Thank you very much.
So we started with the Hollywood presentation, and we ended with the Lego movie. And now we'll have some questions.
Thank you.
Here?
Okay.
So, intriguing that the proteasome is sort of like the homeostasis of amino acids. I was wondering, did you entertain the idea that inhibition of the proteasome may do something to biosynthesis pathway of enzymes? I mean, something that come to my mind like ODC, the orthonine decarboxylase, which is a natural substrate and also synthesized polyamines.
So there's yes, we've looked at this, and there's a whole part of this study that I couldn't present here, but so downstream of the integrated stress response has actually two facets. One is to decrease protein synthesis, global rates of protein synthesis, as I said, to spare the consumption of amino acids when the cells are starving. But in parallel to that, there's also a transcriptional program which consists in induction of genes that are actually very important for amino acid metabolism and amino acid transport. And it was very interesting to find that actually, so in mammals, there are two amino acids that become particularly critical when the proteasome is inhibited. So these are cysteine and asparagine, and it turns out that downstream of the integrated stress response are genes that are the predominant genes controlled by the integrated stress response are genes controlling asparagine and cysteine biosynthesis. So that argues that there's been an evolutionary pressure to really make sure that the abundance of these two amino acids in particular are kept above a certain level.
And kind of to follow up on that, so in the UPR, it makes sense to turn on the proteasome, right? There's usually a folding stress, and there's a bunch of trash that needs to be taken care of. So induction of the proteasome there, it conceptually makes sense. But in the case of inhibition of TOR, you said it wasn't surprising, but I think it's actually quite surprising that the proteasome would be upregulated, because the whole idea is under starvation, you want to get more amino acids. And I understand that getting more proteasome is going to give you more amino acids, but it's also going to consume a ton of amino acids, because the proteasome is pretty big and you're making a lot of it, and it's going to take a lot of amino acids. That, and coupled with the fact that the bulk of the amino acids you're going to get from degradation of proteins is going to come from new synthesis. and synthesis is shut down too. So you're making something that's to get more amino acids, and the math I'm not sure makes sense.
I think it does, because you're switching this. I mean, you're switching the cells. It's not like you make more proteasome, but at the same time you decrease production of other, massively consuming protein complexes, in particular the ribosome. Okay, so you turn down ribosome production to make more proteasome to resolve the stress.
Two more very quick questions, and we have to proceed.
Thank you. It was a great talk. I was wondering about the speed with which the assembly mechanisms affect proteasome. So if you were to track, like by native gel, looking at assembled proteasomes, do that correlate with gene levels, or?
Yeah, we haven't looked at this. It's tricky. I'm just gonna give you, my feeling about this. I think so. I think echoing what Chris said in his first lecture about robustness, if you knock out one single proteasome assembly chaperone under normal condition, you see no phenotype, right? The idea is that these chaperones, I think, provide a kinetic advantage to making more proteasome more quickly to help cells adapt to the challenge. But we haven't measured that.
One thing that I find very interesting is that you have two amino acid sensors, basically. You have the TOR system and you have the proteasome.
Yeah.
But they discover different amino acids. So the proteasome responds to cysteine and asparagine, and TOR responds very strongly to leucine and arginine. So do you have any comments on this? Is this on purpose evolution?
Yes. I think it's coming up in the TOR pathway clearly. It's more than leucine, right? And I think so it's probably not black and white. It's probably not just.
It's very strongly favoring it.
I'm not too sure about this at this point. Whether we should, I'm not sure.
Okay, we need to move on.